So again, just very briefly, American Preclinical is a contract research organization. We're based in Minneapolis. I had the pleasure to be able to introduce or invited to come out and speak to you folks, just to give you a little bit of a sense of not so much what we do, but some ideas about how this idea about streamlining preclinical testing, where you can go at a little different change of venue, change of process than we've had before, not so much a business perspective, but also really looking at ways to optimize processes going on in the more complex medical device programs that we are involved with at our CRO. I'm used to speaker disclosure. I'm not a millennial, just to make sure that's not in question by anybody. Have a few biases of my own, and I am a full-time employee and a part owner in the company. What we want to talk about today, I think, is we'll run through some definitions, a little bit of contrast and compare about the medical device and the drug industry, just in terms of how the system operates and where it goes. And I think, again, something you folks are probably very familiar with. We see it from an awful lot of organizations that come through our process. As I say, we get to play with everybody's really cool toys. It's a great opportunity in the kind of the work that we do, and work with companies anywhere from large behemoth medical device manufacturing groups to what we've already talked about, 2 docs and an engineer, whether those are female or male doctors. It's still just very, very small startup organizations that have a concept and really need to take this thing all the way and take the process all the way through. A little bit of ideas about how those things work from our perspective and what we've seen over the years. Some ideas about outcomes and endpoints of the preclinical testing. Again, these are key components associated with how we demonstrate safety, performance, and we'll talk about efficacy. We'll go into a few details on some specific experimental models that I think are interesting and that have some novel potential, and then just a few take-home messages. We'll just go through these real quickly. Nobody can read these anyway, but the preclinical, basically animal stuff, we work in a majority of our systems, both in vitro processes in the laboratory and in vivo models. This idea about translational medicine, it's really big in the industry and in the country and the NIH and all the granting agencies. Everybody's got a department of translational medicine. This idea, I think, in my mind, and what we tried to do is how well it goes to predictions. And predicting from a medical device perspective, a little different than a pharmaceutical perspective, how well you can make these animal models and other types of systems really predict what happens. It's a challenge. I think we're really trying to get a system where we predict safety, have a good compensation or a good ability to make some general statements about how a device system works and how well it's going to work and how safe it's going to be. And that really might be the limit. This idea about efficacy and effectiveness with respect to a device, and I think we'll talk about the definition here, is that really associated with its efficacy in treating the disease or its performance from the perspective of its engineering characteristics and what it's intended to do. So again, difference between a drug and a device with this respect. Safety, obviously, it's a key component, do no harm. This idea about is that a predictable and basically a translatable parameter that you can understand in medical devices and take the animal models and give you a high confidence that your device is going to be safe in a clinical trial. Performance, again, with respect to a medical device, you have to clearly define the intent and expectations of what that device is intended to do. Is it going to do, what kind of a job did the engineers define for it to perform? How are you going to be able to do that? And again, it's a lot different than an interaction with a receptor for a pharmaceutical or for a small molecule. And then I think we'll touch on the whole idea of biocompatibility in the area of acceptable foreign body reactions. I think buried down in here, there's a quote associated with Buddy Radner, who's been in the biocomp business for a long time. Nothing is truly biocompatible, they just work. And they don't do any more damage. And the foreign body reaction that the human body or that the animal models generate in response to the implantation or deployment of these materials is acceptable. It's an acceptable foreign body reaction. We've already kind of talked about this. Basically, a general slide I always use to talk this up is almost a tenfold difference in how much a drug costs and how much a medical device costs. These are older numbers, but they just give a general concept. Basically, the device systems, there's a lot of people out there. The order of magnitude, so certainly in terms of what the costs are, what the numbers of folks are out there, how much it gets worked with. But the idea is that, we've already sort of talked about this, what really constitutes a functional device, and it's really been intent associated with unmet medical need. Ken gave this really very accurately early on from respect to the conversations we've seen today. the idea that you have to be able to sell this. You have to be able to get the dollars to generate these kind of processes and be able to support the kind of work that we do. Our organization is a fairly expensive process associated with the medical device development process, and you need to be able to generate the funds to be able to basically to afford the kind of work that our organization or any other organization that generates these kind of preclinical trials is able to do. And again, to do that, you have to do all the things that you've heard about today from both IP perspective and from an investment perspective. I'm a Pfizer retiree. I'm not only a non-millennial, but I'm also a Pfizer retiree, so that in the old days when we used to work in this environment, my job as a project leader in that organization was to work on target identification. You go to scientific meetings, you talk to consultants, to thought leaders, to physicians, and find those issues out there that had a potential for an unmet medical need for a pharmaceutical target. You develop some sort of a process associated with what's the molecular activity going on there. And again, you either devise a chemical that attains that goal of an inhibition or an activation of a process, or now a biological that does the same thing. You as a project leader are given a medicinal chemistry team with our cloning and expression group, and you're off to the races to go through a process that's sort of similar to this. You confirm your target identification validation, you confirm with the medicinal chemistry or with a new biological, you keep going down the road. It's a relatively linear process. It's uniform for most of the solid oral dose drugs, which is where most of the blockbusters come. It's changing now because a lot of the new blockbusters have transitioned in the last few years to being biologicals. But again, this idea is that the business model is most similar to a wildcat oil speculation, that you can hit dry holes. And if you do that, your failure anywhere along this line is pretty much start from scratch. And again, my job in the pharmaceutical business always was to kill drugs often and early and cheap. So the further to the left you stay in this process, when you eliminate or shut a procedure down, the more money you save for the company, and you hope to filter out only those programs that are coming through in phase 1, 2, and 3. We could talk forever about why drugs fail and so forth and like that, but just to give you a general analogy, that's what chemistry is associated with, or biology with the pharma business. The discovery idea is a little different. Again, you're looking for a concept or an idea. You try to come up with an unmet medical need. You decide what needs to be built. You come up with some initial design processes. You go through some prototypic design, and you come up with a material that looks like it's worthy of testing. Just schemes off the internet. The process could be look similar. There's a product concept. There's initial product design. There is bench testing and iterative design could feed on to preclinical feasibility assessment, final manufacturing device design, product freeze associated with somewhere along here there's biocompatibility testing, preclinical safety and product work with the final GLP safety assessments, and then you're on to your regulatory submission. But I think it's more like this from what we've seen and what you guys probably experience and what we certainly see in our environment. And I think the idea of trying to come up with these iterative processes, it's different and a little bit more involved with respect to what goes on. And just to break some of these down, I think basically the idea of early stage assessment, looking at biocompatibility and materials assessment here is one real key component. The concept of biocompatibility, of materials assessment, using materials that have been well characterized or used before. Again, sometimes it's nice to come up with a brand new material. The potential and the challenge from that, from a preclinical perspective and a biocompatibility perspective, can be really insurmountable sometimes. Just the extra work and the extra effort associated with a new and novel device. Again, we work with companies that use both routine materials, standardized materials. They understand those arguments are always in place. But then we also have companies that come through us that have, gee, I have a brand new novel material. Well, you're in for a wild ride. I mean, you might have a characteristic that's really going to be unique and maybe satisfy and do some unique things with respect to the efficacy and the performance, but it's going to be a challenge from a regulatory perspective. There's no question about that. So we're understanding more and more in our system about how we work on benchtop testing of models, how we work on with ISO guidelines, the newer concept with the FDA and understanding the chemical characterization of these materials, their extractables and leachables, and predicting from that what exposure characteristics are going to be and what toxicology and toxicological risks are going to be. And then there's quite a bit of work in vivo biocompatibility as well. It's not on this. The middle segment, we'll talk a little bit about this in a minute in terms of selecting the right kind of animal models for feasibility. Again, understanding, is your device doing what you think it's going to do? How well can you characterize that performance aspect of a device? And can you use existing established models? Do you build your own models? And I think the key component here is that safety prediction. What is your specific path to establishing and generating a high probability to predict that your device is going to be safe in patients? And what animal models can you use to assure that? And then I think the final component is really the ultimate design of those studies, using existing and available FDA guidance documents, making sure that you achieve the right study duration per the FDA guidance documents or per your pre-IND meeting or your pre-sub meeting. What are the safety endpoints that you're trying to attain, and what are the function and performance assessments? And again, you'll notice here, I don't have an efficacy component here. Within medical device business, efficacy is still, we almost look at it as a dirty word. It's just a hard process to really demonstrate and prove. It can be done. You're ultimately going to generate that information in the clinic, and what can you do from a preclinical perspective to really assess that? We'll talk a little, just the new guidance documents are out from the USDA, or from the FDA in last year, middle of last year, and you can't read these, but basically this is the kind of a complex matrix that you're supposed to be able to read, and our groups like us or our colleagues at NAMSA or the other folks in the industry environment associated with biocompatibility. This is basically almost a commodity. The pricing is such, the turnaround times are there, and everything is really built for checking the boxes and making sure that you get your biocompatibility endpoints appropriately. And again, the agency is coming out with a new interpretation of the ISO guidelines. They're not exactly the same, and there's just enough difference there to probably really irritate and make a lot of people frustrated in the industry. And the guidance that we were able to offer, and our colleagues like NAMSA and the Wuxi and the other folks that are in this business, it's frustrating for us to try to understand and predict what the FDA is going to want, how closely they're going to follow the ISO guidelines and so forth. But one of the things that really is coming out, again, this is a standard panel of in vivo assays in addition to the standard in vitro processes. But a new process that's really coming down, I think, these days is this transition from in vivo to in vitro. The idea about minimizing animal use. There's always a process by which you can at least attempt to design and build laboratory systems using cell culture systems or other processes that can significantly reduce both the utility, the predictability, and the feasibility and reproducibility of these assays. Many of these old in vitro assays that I just showed you are from back when I was in graduate school, or sometimes even worse. And it's kind of almost, we really don't like them. We hate doing those assays. The guinea pig maximization as a standard assay for sensitization has been around for a long time. It's a box check. It keeps people on the ISO board as well as the FDA happy, but there are newer systems that are coming out that I think really can come up and help predict these systems, predict these parameters with a higher probability to really, again, predict what the ultimate safety is of your devices. We'll just look at a few of these. Again, some documents and some newer procedures that are out there. I just popped this one up this morning. This is new from in vivo to in vitro, and that's really a consortium of many of the logos. I left out a few academic institutions. Medtronic, NAMSA, Eurofins, Eintox, J&J Medical Device, Hopkins, the FDA are supporting. They're represented by authors on this paper, but there's an idea about actually putting some teeth into this transition from medical device testing through in vivo models into the in vitro systems. If you look on the USDA website, there's a classification for animals that are tested and potentially without or with minimal treatment for pain and distress, and that's part of what the guinea pig maximization requires an animal to be scored at. And all the medical device testing labs are out there hanging on the USDA website with a very high score for their total guinea pig utilization, and it makes people a target for a lot of the animal rights groups. And it's unfortunately, it's a standard that's brought by managing the ISO 10993 guidelines, and the agency and the ISO groups require it. But these are really some significant attempts to try to transition from those animal models into the in vitro system. So again, as a newcomer to the medical device biocompatibility testing, it's been a goal of APS, and I think a lot of our colleagues to try to cut that animal usage down. There's another test that's been around for a long time, the idea of hemocompatibility, specifically thrombogenicidity testing. Anybody who's made a catheter-type device or any kind of long, skinny medical device that's going to be exposed to human blood or to blood in a patient has looked at these. There's an anticoagulated venous model or an implant model. Basically, it takes a device, your device and a competitor's device that's legally marketed. You put them in a dog, you let the animal cook for 4 hours, you take them out, and basically it's a sacrifice of the animal. There's no way to get around this without actually doing termination. You look just for thrombogenicity, how well the device is going to react when it's exposed to blood in that model. And this is actually required by the agency, even for materials that are going to be used in coagulated environments that are catheters that would be typically used in a setting where a patient would be heparinized. So it's kind of an archaic process in the old testing scheme. We've actually worked internally to try to come up with a way to do this with sheep blood without sacrificing animals. And we've made some progress on that, just a little component of what APS is trying to do. And again, to try to reduce animal usage and try to come up with a better test. Not only is it a non-animal sacrificing assay modality, but it also has the ability to really bring statistics and assessment into this thrombogenicidity testing, which is a bugaboo for many of you folks in the medical device business. Just a little bit about preclinical models in general, and I'll kind of reiterate this. The materials assessment for both in vitro systems that we've worked with, you're still always going to be implanting these devices into healthy animals. The limitations associated with tissue response is still kind of wound up into that idea. Safety, again, we do assessment of medical devices in healthy animals. If you think about when we got our start early on, it was working with some major stent companies that were in the Minneapolis area back in 2006. And we put 2 to 3 coronary stents per animal into what was basically a toddler pig. So these are pigs that are 4 or 5 months old, maybe 2 or 3 months old. They have extremely exquisite looking coronary arteries, and we put a stent in and wait for it to sit for 3 months to 6 months to 9 months. And then we take those animals down. We evaluate the histologic response to that, and then that's what we use to make our safety predictions. So is it a flawed model? It's got a lot of potential, and again, its predictiveness has been pretty good. Systems that really had a problem in that sort of a simple model have shown to be problematic with respect to clinical trials too. But remember, we're always looking and working with healthy animals. We'll talk a little bit here about some of the transgenic or bred models, some other options that are extremely elegant, and they have their limitations. I think they have potential for some uses. Depends upon what your endpoint is, and I think as we talked about earlier this morning, it depends upon who your audience is. If you're trying to really generate and get to a regulatory submission, get to the information that you need to go in first in man, to get to an IRB, or if you're trying to convince your investors, or if you're trying to publish manuscripts and papers on the system, there's a lot of diversity in terms of what your endpoints might be and what kind of models you might want to look at. This is, I promised our business development guy would show this slide because this is an obligatory APS commercial break. And it simply shows that if you go to laboratories that use a lot of animals, number one, we're trying to reduce our numbers here, but it's an indication that there's an awful lot of animal model development. This basically is just a chart of numbers of animals used over the years versus APS, which is at the top of the list right now. We don't have some of our exclusive medical device clients on here. MPI is another good example. These guys are through the roof with respect to animal usage, and Covance and some of the big players. But we tried to just compare ourselves with where some of the smaller contract research laboratories that specialize in medical devices are. So we go through, this is an inclusion for sheep, pigs, dogs, and cows. So again, lots of animals go through our laboratory on a basis, as well as do our competitors. So a lot of animal work is being done here, hopefully all for good purposes. So let me talk a little bit about some specific models that I think have really been developed by some of our collaborators and some colleagues that we're working with in different parts of the industry. An awful lot of medical devices target diseases associated with hypercholesterolemia or atherosclerosis, obviously, from a medical device perspective. A lot of endovascular processes. There are some really interesting systems out there. And one of the best ones, just to use as an example, how do you get an atherosclerotic lesion? How does one generate a target lesion in a coronary artery or in a peripheral artery or an aorta that's prone to aneurysm? Many of these systems are obviously big targets for the medical device community. how do you generate, with some kind of reproducibility and some modest and reasonable expense, an animal model that can generate those kind of lesions for you? Again, if you want to pursue that sort of a target. Well, this is just a very intriguing group. EXEMPLAR Genetics, it's another one of our northern colleagues. They're based in South Dakota. We've collaborated with them over the years, and basically they generated an animal with transgenic work where they did a knockout of the LDL receptor. So basically this animal has familial hypercholesterolemia. Cholesterol levels are in the range of an unmodified animal, or in the range 300 to 450 or so, versus 350 to or 500 or 50 to 100 for the normal range. Just that's total cholesterol. So the animals are very much prone to generating plaques. And again, you can't see the pictures too well here, but those are ugly lesions. That should be a nice round circle, and it's not. That's probably what my arteries look like, but that's the sort of situation that you can generate in these animals. There's a Watanabe heritable hyperlipidemic model too that's also out there. The problem with these guys is they're rabbits. They're still small. You've got an aorta to work with and iliac arteries, but with these swine that you actually can use, this is bred into a Yucatan mini swine, so the size and shape is appropriate for these animals. They don't gain a whole lot of weight over their lifetime. You can catch them at a year and a half of age, and they have a potential to demonstrate some of these significant lesions. Problem with this is they're elegant models for pharma. The idea about managing and inhibiting that process of hyperlipidemia has been great. They've been a nice, great confirmation. Pfizer ran a big study on this using statins, really showed an extraordinary and exquisite, again, very consistent with human data. The kicker is with most of the statins, you've already got quite a bit of human data available out there for you. So both the swine and the WHHL rabbits both are very, very nice for this models. The problem is that despite what you try to do to these animals, you can put them on accelerated chow, you can do a variety of things, you can go in and damage the vessels and generate what you think would be sites predisposed to generate these nice atherosclerotic lesions. But you essentially have about a $6,000 pig to start with versus $100 or $500 for a domestic swine. But you've got a $5,000 or $6,000 pig with his licensing and all his IP. And then you potentially have to put him on a special diet for as many as 6 months to 9 months. And you still don't have as high probability for assured generation of lesions in these animals as you would like to. So it's been just a difficult process. So the idea is still of treating an athero disease state with stents, with any of these other kind of slick, again, the fun devices that we get to work with are a little bit more complex and invasive medical devices, angiography, drug-eluting balloons, atherectomy systems, and any other processes. This model, just although it had potential, I think it's very elegant, it just simply doesn't really give you the bang for your buck that you need to. And it's very expensive. The other question is, does anybody in the medical device community want to test all their stents in a $6,000 or $7,000 or $8,000 animal that might still only have a 1 in 5 chance of having, or 1 in 3 chance of having a good coronary lesion? So elegance was great, but a process that we put in place here, and it's just a simple system. We actually would go in and create a plug of femoral bone marrow. Essentially, it's a calcified section taken out of the core of the bone marrow. Process it, we can get sites that are anywhere between 2 to 6 millimeter in diameter. We can generate these with a lesion or a hole, some kind of a lumen in them. We can deploy these in animals. We can put them in the coronary artery, 1 or maybe 2 in a coronary artery. We can put up to 4 sites in a peripheral artery. And we can basically generate then in a very simple method, in a simple way, in a relatively inexpensive way, in a domestic swine of appropriate size, because you typically use a big pig for this, but it's still a relatively expensive or relatively reasonable model from an expense perspective. And you can get up to 4 peripheral sites or 1 or 2 coronary sites per animal. And you can actually work on these. We can prep these animals in the morning, and you can come in and do your testing in the mid-morning or the afternoon. On angio, they look pretty nice. You get a nice size stenosis in both the coronary or peripheral arteries with a known lumen size that can be predicted in the right site. This is just a slide that was presented by some of our colleagues at Riva several years ago, where they actually used the site to demonstrate you can make the lesions soft or hard by a pre-treatment in a decalcification solution. So the lesions on the left that were too hard really deformed the stents on its deployment, and you can see the lesions on the right were able to be fully formed and actually modify and modulate the size of the calcification spot. You can see that in the system. But they're good for either acute or chronic treatment processes. Again, you can put balloons in these animals. You can go stent over these devices to get a sense for how the stent's going to feel in an otherwise, it's not truly, from a pathological perspective, from a cellular perspective, it has a lot of differences between a coronary lesion. But from many perspectives with respect to the physical size, shape, and the characteristics of behavior with respect to medical device performance assessment, it really does a pretty good job. We've had a lot of physicians that have deployed stents into these, and they say, that feels just like the patient that I stented last month. So I think it really does work from that perspective. From drug-coated balloons, you can actually deploy balloons in this calcified lesion. You can look at differential site, differential uptake on the balloon, the effect of that sort of a process. You can check for flow pressure. It's a guaranteed stenosis. It's not something that requires any specific amount of time or the probability that, gee, only a third or two-thirds of these treatments that we use generate a calcific lesion in an animal. You can use them for crossing wires, for specialty catheters. You can make them complete occlusions as well, too, with appropriate softness. So that's just a nice way to kind of customize a process. And atherectomy, it's a great opportunity for both mechanical and other types of atherectomy devices. Physicians, again, who've looked at these and have treated them with atherectomy processes say this is great. It really is exactly feel like a clinical case. I don't make this really to try to sell the lesion, although it sounds like it is, but more than anything else, I'm just trying to show that this idea about designing a system that is truly associated with performance and safety is really critical to the outcome that you're really looking for to try to advance your process through that complex scheme that I talked about before, and not something that's really going to be elegant and show how the efficacy of your device would be in a model of a true atherosclerotic lesion that has similar characteristics to what a human would be. There's hypertension models that are similar, again, in a very similar process. I don't want to drag on this too long, but the idea is that there's an Ossabaw swine species out there. These are animals that were derived from a colony that was found on an island in South Carolina. When you Google Ossabaw on the internet, it's interesting. Half the hits come back as medical characteristics, and the other half come back as recipes, because these animals are extremely well marbled and have very, very tasty pork chops, apparently. But the idea about this is a model. Again, it's a very elegant model for hypertension, for transition into type 2 diabetes. There are processes now that they're looking at cloning programs underway to really get these guys either with an LDL knockout to combine their natural predisposition for hypertension and type 2 with elevated hypercholesterol. Again, just quite a bit of work and quite a bit of energy and quite a bit of expense going into these models. They're great, they're elegant, but I think the utility of these systems is really difficult unless you're trying to really demonstrate efficacy. Efficacy is hard, there's just no way around it. There's some even more complex ways that you can basically kill the islet cells in pigs just as you can. You can do this in mouse or rats or any other system for looking at glucose tolerance. These are a little bit faster, they're quicker, and they get you to two-thirds of what you want to with respect to some efficacy assessment in the device community. But still, there's limitations in terms of what these processes can do. One of the, and I have to, although I'm preaching against this, I really have to say this is, to me, is one of the coolest and most elegant processes. So again, I've been in this business for a while. I think this is actually, you can't really read it, but that's a paper that we published in 1980 on treatment with immunotherapy studies in mouse tumor systems. So obviously it's only been relatively recently that this has happened, not back in the 70s or 80s when I was working on it in graduate school. But understanding how these models of treating cancer work and what you can do with respect to understanding proof of concept for biologicals, that's a whole other area. The device characteristics for looking at tumors in treating cancer, there's quite a bit of therapeutic systems for surgical processes, for treatment processes which involve energy delivery, just a variety of systems associated with medical devices and tumors. And again, our friends at EXEMPLAR Genetic have developed an inducible tumor that you can use with an injection of a local inducing agent at the site of the tumor that you want. You can make a liver cancer, you can make kidney cancer, you can make squamous cells, you can make muscle. So it's been a very, very interesting target. I think this is a really exciting area so that you can then, in a large animal, in a swine, a mini swine, actually target and induce a liver tumor or hepatoma, a lung cancer, a renal cell carcinoma And it would be, again, it's not exactly associated with the natural progression. It's sort of as elegant as what we were even doing back in the mouse models in the old days. But a target then for diagnostics, to assess CT efficacy, to target selected markers for certain of these things, just to look how these would work with imaging processes. Tumor-specific contrast agents would still be functional in these kind of models. Could you go in and do surgical resection of these and then look at the aftermath of that? Can you do other kinds of imaging? Can you do processes? Can you look at adjuvant chemotherapy? There's lots of work now with drug delivery systems and with embolic devices that both occlude as well as take out systems and really reduce blood flow to these tumor sites. So again, the key thing is just many of these elegant models are associated with where we are with what you can do in the tumor models. We'll walk through these really pretty quickly, that the preclinical models come in a wide range of shapes, sizes, and complexities. The drugs work through their inherent characteristic of biochemistry. Devices really work on essentially what the engineers have decided and how they've designed them to perform. We've already talked about this, know your audience and your customer when you're looking to really try to define the preclinical program and match the complexity of your models to the endpoints. I think the key component here, really the endpoint, is the elegant and exhaustive may be the enemy of adequate and definitive. I think some of these very nice, elegant models are just not going to get you where you want to. They're going to cost a lot more to do. And I think the endpoints associated with satisfying regulatory bodies with some of the more traditional and standard models is really very adequate and very easy to get to. Again, this idea about safety and performance are much easier than efficacy and effectiveness. The last two are really sometimes almost impossible in the medical device world. And again, I think this idea about translational, I think the key component for translational activities is, can you take a model and use it to really safely and with high confidence predict human safety in your clinical trials? And that's really what our main goal is, and the main goal of most of these preclinical models. And that's a wrap. I was given a signal from the back, so we'll try to keep it low. I appreciate everybody's time and attention.